Researchers from the University of Pennsylvania have made a groundbreaking discovery that red blood cells, rather than merely acting as passive participants, actively contribute to the contraction of blood clots. For years, the scientific community maintained that red blood cells simply accompanied platelets during clot formation without any significant involvement. The new study reveals that red blood cells play a vital role in tightening and stabilizing clots. This discovery transforms our understanding of one of the body's most critical processes, remarks Rustem Litvinov, a senior researcher at the Perelman School of Medicine, and co-author of the study. It paves the way for innovative approaches to studying and potentially treating clotting disorders, which can lead to excessive bleeding or hazardous clots, such as those associated with strokes. Published in Blood Advances, the research challenges the long-held view that only platelets, small cell fragments essential for sealing wounds, were responsible for clot contraction. The Penn team illustrated how red blood cells directly aid in fortifying and stabilizing clots as they decrease in size. Red blood cells have been studied since the 17th century, notes co-author Prashant Purohit, a professor in mechanical engineering and applied mechanics at Penn Engineering. The surprising reality is that we continue to uncover new insights about them in the 21st century. An unexpected finding previously, researchers attributed the process of clot contraction solely to platelets, which pull on the fibrin protein strands to enhance the tightening and stabilization of clots. As co-author John Weisel, a professor of cell and developmental biology within ITSM, and an affiliate of the bioengineering graduate group at Penn Engineering, explains, red blood cells were perceived as passive bystanders, merely assisting the clot in achieving a better seal. This assumption crumbled when Weisel and Litvinov conducted an unexpected test. They created blood clots without platelets and anticipated no change. To our surprise, the clots shrank by over 20%, Weisel states. They confirmed their findings by repeating the experiment with regular blood treated to inhibit platelet activity, which still resulted in clot contraction. That's when we recognized that red blood cells must be doing more than just filling space, Litvinov adds. Modeling the mechanics of blood clots to understand how red blood cells facilitated this unexpected behavior, the team sought assistance from Purohit, whose mechanical engineering background proved invaluable. This process parallels how particles in colloids, such as paint, milk, or muddy water, aggregate and form clusters when their surrounding conditions shift. The proteins in the surrounding fluid create a pressure imbalance that pushes red blood cells together, explains Purohit. This attractive force causes them to compact more tightly, contributing to clot contraction even in the absence of platelets. How clotting works without platelets. As blood begins to form clots, a web-like protein called fibrin creates a mesh that ensnares red blood cells, drawing them closer together. This packing sets the stage for osmotic depletion forces to take over, Purohit asserts. Once red blood cells are tightly grouped within the fibrin mesh, proteins in the surrounding fluid are expelled from the narrow gaps between the cells, resulting in a higher protein concentration outside the packed cells. This balance creates a difference in osmotic pressure that acts like an external squeeze, pushing the cells closer together. This attraction causes the cells to aggregate and transfer mechanical forces to the fibrin network surrounding them, adds Purohit. The outcome is a stronger, denser clot, even without platelets. Validating the model, previous research hypothesized another explanation called bridging, where small molecules on the surface of red blood cells attract them to adhere. Our model acknowledged the bridging effect but indicated it was significantly less influential than osmotic depletion, says Purohit. To test the model, first author Alina Peshkova, now a postdoctoral researcher in pharmacology at PSM, performed a comprehensive series of experiments on modified blood clots. In the absence of bridging molecules, the clots still contracted, but minimal contraction occurred in environments designed to obstruct osmotic depletion. We experimentally validated the model's predictions, Peshkova states. This is a prime example of theory and practice reinforcing each other. Fighting clotting diseases and strokes. A deeper comprehension of the role red blood cells play in clot formation and maturation may lead to innovative therapies for conditions such as thrombocytopenia, characterized by low platelet counts that result in uncontrolled bleeding. Furthermore, these findings could enhance our understanding of how clots can fragment and travel through the bloodstream to create blockages, or embolisms, which can trigger strokes. Ultimately, our model will aid in understanding, preventing, and treating diseases related to clotting in the bloodstream, asserts Purohit. Reference: red blood cell aggregation within a blood clot causes platelet-independent clot shrinkage by Alina D. Peshkova, Ekaterina K. Rednikova, Rafael R. Kazmatulin, Oleg V. Kim, Vladimir R. Muzikantov, Prashant K. Purohit, Rustem Litvinov, and John W. Weisel, the 10th of July 2025, Blood Advances. Thanks for watching. If you are new to our channel, please subscribe to our channel and turn on the bell for further notifications.